Hello students, in this video we're going to do lab 4 together. So let's go to D2L and look at the instructions for lab 4 right now. The instructions for lab 4 enzyme function starts here with goals and part 1, setting up the enzyme digestion. So here are the materials that you need. You need 3 ml plastic pipette from lab 3, which is this plastic pipette. That's what it looks like. And then you also need this 50 ml blue cap tube. You need total of 5 of them. And one of the blue cap tube will label lab 4, and which has all the materials you need in them. Okay, and then within this lab 4 tube, you will have the lactase in it, the lactose, and of course you will need a thermometer from your lab kit. This is what it looks like. And you will also need your micro pipette and your the tips that is within this lab 4 blue cap tube that will fit onto the micro pipette here. And then you will also need 4 any kind of container. It can be cups to hold water, to keep a water bath, to incubate the reaction with. So let's go into the procedure and see what we're supposed to do. So the first thing you need to do, like I said, is you should take out this lab 4, upside down, sorry. The lab 4 tube, which has a lot of stuff in it. For example, it has this micro pipette tips that you're going to use with the micro pipette. We're going to put it on the ends of it. Make sure it's secure. Okay. You will also need to, you have 2 of these micro pipette tips. And for these tips, you have to reuse them by rinsing them in tap water. And then so the first step is to take out your lactase tablet, which is in this little guy here. So what I would suggest you to do before you're opening it, you should crush it with the back of a spoon or a knife, whatever you want to do, or a hammer. So I get a regular knife, and then you will put this on the table before you're opening it, and then you just gonna crush it. I hope you can see it. You just put it on the table and you're gonna crush it before you even opening it. Okay, and then you can feel it, see if it's crushed. If it's not, you can just use your hand to break it even more, and if not, you just do a little bit more. After you crush the tablet within the package, now you can safely open it. Okay, after you open it, what you want to do is take one of these blue cap tube, remember? And then that you should have seen, just pour this whole package in it. This way you don't have to make a mess, otherwise it will fly everywhere. Then it's not going to be pretty. Okay, so then you want to make sure label your tube. Lactase enzyme. Okay, so the next thing you want to do is get some tap water, what you have at home, and you can see that this tube has marking on the side. So find the marking that says 10, which is right, the way I look at it is right here, 10, and you pour water in it until it hit 10. Okay. Close the lid securely. Once again, you will hit the line that marked 10. Give it a little shake. Okay. And now you have it labeled lactase enzyme. Let it dissolve. Put it aside somewhere. Waiting for the lactase enzyme to dissolve, you should label 4 other blue cap tube. 2 to 5. So you see this is tube 5 and tube 2, right? You can use your Sharpie or the crayon that comes with the kit. But anyway, all 4 tubes need to have a number on it. 2, 3, 4, 5. I want to show you quickly before you lose sight of what we're trying to do. So we're trying to set up table like this. We have 5 tubes all together. You have 1 tube with just the sugar lactose. which I'm going to show you how to make in a minute. And then the other 4 tubes will have the sugar lactose plus the enzyme lactase. That's the one we just crush and mix with water. So the goal is to look at whether this enzyme lactase can work on the lactose to release monosaccharide for the yeast to ferment in part 2, okay? So keep in mind this is what we're trying to do, and then it will all make sense. And then not just that, you see that tube 2 to 5 is identical, right? They all have lactose and lactase, but the difference is the temperature we're going to put them in. So one of the tube number 2 will be in room temperature water bath, and then the other 3 tubes will be in different temperature, 40 degrees, 60 degrees, or 80 degrees. So we're testing how different temperature affect enzyme activity in this way. Now, based on the table I just showed you, the 4 tubes you just labeled, tube 2 to 5, we're going to all add lactase in it. You know, the enzyme you just make. It's not going to dissolve completely. It'll be kind of milky, chalky color, but that's okay. So you're going to take your micro pipette, not your micro pipette, your plastic disposable pipette, and if you look carefully, you will see that there's marking on the side of the disposable pipette, and it has 1 ml, 2 ml, and 3 ml all the way to the top. So what you want to do right now is to get 1.6 ml. So it's just a little bit above the mark between 1 and 2 ml, okay? So let's do that right now. We're going to take 1.6 ml of this lactase enzyme that you just make. Because it doesn't dissolve completely, you make sure that it mixes well before you take out 1.6 ml. And look at your micro, you want to suck it up, and then you hold it with your finger, and then it doesn't have to be exact, approximate is good. So it's the mark between 1 and 2 ml, and it's just a little bit over it. And then when you do that, and then you put it in tube 2, and then the rest of the tube, okay, tube 3, tube 4, and then we're going to do the last tube, tube 5. So remember from that table, tube 2 to 5 all have the same thing. They all have lactase. They all have the sugar lactose enzyme and the sugar, the substrate. So now we are ready to set up tube 1. Same thing, you take a blue cap tube, label 1, and instead of the enzyme lactase, what you're going to do is you're going to rinse the plastic pipette out with clean water because you don't want to contaminate it with enzyme, and just use that to put some regular water, tap water, same amount, 1.6 ml, just like the enzyme, into tube number 1, which don't have any enzyme. Okay, so make sure you do that, and make sure you rinse the plastic pipette. Don't want any contaminations. Okay, now we're ready to use the lactose that looks like white powder from the Lab 4 blue cap tube. Remember that it has like micropipette tips, have yeast, and then this is one of the white powder. If you don't remember what it looks like, go back to your lab kit packing list. It will show you exactly. But basically, it's a small micro centrifuge tube with white powder in the bottom that is inside the Lab 4 50 ml tube. You want to open up the cap, and you're going to use the same plastic pipette that you just used to transfer water to Lab 1. And now you want to take 2 ml of water. So you will take water, 2 ml. Look at the mark on the side of the plastic pipette, and you want to add it to this tube. It should go all the way almost to the top. And then you close the lid, give it a shake. Do a little flick. And then let it sit on the side, let it dissolve a little bit, okay? So after put the water into the lactose powder, remember lactose is a sugar, it's a substrate, you're going to test it on. You should write 10%, upside down again, 10% lactose on the tube so you don't get confused what this is. And it should dissolve somewhat, but if not, completely, it's okay. So right now what they want you to do is to take your micro pipetter, put a tip on it, make sure, okay, and adjust the reading to 200 microliters. So it should read, oh, get closer. It should read 200, okay? And when you pipette it, remember the way to do it is you use your thumb to hold this down to the first stop, not to the second stop, okay? Try it again. Hold to the first stop, not the second stop, okay? The second stop is for ejecting all the stuff. So once again, you take your lactose sugar solution you just make, give it a shake again. Open the lid. You want to hold your pipette to the first stop, hold it that way, and then go into the solution. Suck it up, and when you set it correctly, it should look almost to the top of the tip, about right here. Okay, so make sure you set it at 200, and then now you're going to put 2 times. So we're going to put 200 in each one of the tubes. So let's put one here, and we're going to do that twice into each tube. Give it a shake each time because sometimes it settles. And then same thing, hold to the first stop, take out 200 microliter, which should be right about here, the liquid. And then take your tube. So each tube should get 2 shots of this. Okay, so let's go ahead and do the rest. So I just took 2, but you can start with tube 1, and then all 5 tubes should get 2 shots of 200 microliter of the lactose sugar. Okay, so after you finish putting the lactose in all 5 tubes, so now is a good time to double check. If you make a mistake, I'm sorry, this is all there is enough for just one time setup. So you really want to watch this video, make sure you know what you're doing. So this is what you have done if you have done it correctly. So for tube number 1, all you have is the sugar solution, the 2 shots of lactose you put in. And in the other 4 tubes, you should have sugar and the enzyme. Okay, so now we need to put these tubes into different temperature right now. So all you need is to use a microwave to boil some water. So that will help you to adjust the temperature to 40, 60, or 80 degrees. So let's take a look at what you should do. Now then you should have 5 containers. Okay, so what I use is just regular plastic cup. You see what I do here? So I label 2 of them RT. Where is it? RT means room temperature. So all you have to do is put tap water in it. And after you put tap water in it, you use your thermometer to measure what is your tap water temperature is like. Okay, once you've done that, you need to write down that temperature in this here. You see RT equal, so you need to write down what is the temperature of your tap water here in this table. And then after you fill it like halfway with water, the goal is, if you worry about how much water to put in the cup, the goal is the water need to be able to cover up the amount of solution that is in this blue tube. Okay, so I would say if it can cover up, if the solution is about here, you just need enough water to submerge the whole reaction underwater. So it can be half a cup, can be even a quarter of a cup, because it doesn't look like there's a lot in here. Okay, but to be safe, just to be sure, you have about half a cup of container of water, and then you will put, actually you only need 4 containers because you can put tube number 1 and 2 into the same container. So here's my tube number 1 and 2. I put in the same container. Okay, so then you can save 1 container. You don't need 2 RT container. But anyway, you write down what is the temperature of the room temperature tap water, and then this is done. You let it sit for 30 minutes. Now let's do the other temperature. So like I said, I already have cups labeled 40 degree. So what you need to do is, you can fill this with tap water, but I would recommend to be a quick way is not to fill with cold or room temperature tap water. You can start with a warm or even hot tap water, and then you can adjust with either hot water or cool water until you reach 40 degree. Okay, so you can start with something warm, and then you will use the thermometer to double check, make sure the temperature is correct, and then you put your tube number 3 into 40 degree, following that table I show you, and then you will do the same for the rest of the other 2 cups. So this is 60 degree, and I will start with something warmer, even warmer than the 40 degree tap water, and then put your tube number 4 in it. Okay, and then the same thing for 80 degree. Okay, and then what you want to do is, after you set the temperature with the thermometer, make sure it's all correct. Every 5 to 10 minutes or so, you want to use your thermometer to keep track of the temperature, because as you can imagine, warm temperature like this will gradually go down. So when the temperature decreases significantly, you want to keep adding hot water to keep this water in the cup at a constant 80 degree for 30 minutes. Okay, so then after 30 minutes, then you can remove the tube, the blue cap tube, from the cup, and it's ready for part 2 of the lab.